There is a moment that happens in veterinary genetics laboratories that researchers almost never talk about in public, and it is this. When scientists sequence the genome of a green-eyed cat and then look at the corresponding genetic data from a human with green eyes, the molecular switches involved are so structurally similar that the comparison stops being surprising and starts being genuinely unsettling. Two species, tens of millions of years of separate evolutionary history, and yet sitting at the same genetic crossroads, pulling the same biological levers, arriving at almost the same color through almost the same molecular logic. If your cat has green eyes, you're not simply looking at a pretty face. You're looking at one of the most striking examples of convergent evolution ever documented in vertebrate pigmentation science, and the story of how those eyes came, with what green actually is, because the answer is not what most people expect. There's no green pigment inside a green eye. Not in your cat, not in a human, not in any mammal that has ever been studied. What looks green is a physical illusion produced entirely by the interaction of light with tissue, and understanding that illusion is the first step toward understanding everything that follows. The iris of a green-eyed cat contains a relatively low to moderate concentration of a pigment called eumelanin, the same brown-black pigment responsible for dark coloration across virtually every mammalian tissue, from fur to skin to the back wall of the eye itself. When eumelanin is present in small amounts and distributed across a translucent fibrous tissue called the stroma, something unexpected happens to incoming light. The shorter blue wavelengths scatter outward and forward through a process called Rayleigh scattering, the same physical phenomenon that makes Earth's sky appear blue. The longer red and yellow wavelengths pass through and are absorbed by the pigment layer behind the iris. What reflects back to an observer standing in front of that eye is neither blue nor a literal physical sense, wearing a color that does not exist as a stored biological material anywhere inside its own body. The color is generated molecular story of how a cat's genome arrives at exactly the right eumelanin concentration to produce that specific optical effect. That is where the science becomes genuinely extraordinary. Domestic cats, Felis catus, have been living alongside human beings for somewhere between 10,000 and 12,000 years, a timeline that aligns closely with the earliest sedentary agricultural settlements in the Fertile Crescent, where grain storage attracted rodents, and rodents attracted the wildcats, who were already beginning the slow, largely self-directed process of becoming something new. For most of the 10,000-year relationship, humans exerted almost no deliberate selective pressure on cat coat or eye color. Dogs were bred intentionally and extensively for behavioral and physical traits, almost from the beginning of domestication. Cats were not. They bred largely according to their own preferences, which means that the eye color diversity we see in domestic cats today was not engineered by human aesthetics the way it was in dogs or livestock. It emerged through a combination of natural selection, genetic drift, and the specific population bottlenecks that happened as cats spread out of the Near East and followed human trade and migration routes across the ancient world. A 2017 study published in Nature Ecology and Evolution, which analyzed ancient cat DNA from archaeological sites spanning 9,000 years and 3 continents, found that the domestic cat's global spread occurred in 2 distinct waves. The first moved out of the Near East with early Neolithic farming communities. The second, somewhat later wave, moved across much of the ancient Mediterranean world along maritime and overland trade routes, likely accompanying grain shipments on Phoenician and later Roman vessels. What this means for eye color genetics is significant. Every major population of domestic cats alive today carries the genetic legacy of those 2 dispersal events, including the founder effects and genetic bottlenecks that occurred when small groups of cats, carrying only a fraction of the total ancestral genetic variation, established themselves in new regions far from the original source population. Green eyes, in this context, are not a single mutation that appeared once and spread outward the way blue eyes did in humans. They are the predictable output of a specific range of pigmentation gene combinations that can and do arise independently in populations wherever the underlying allele frequencies happen to land in the right zone. The primary genes involved in feline eye color have been progressively identified over the last 15 years. Research published through the Feline Genome Project and subsequent studies from institutions analogous to the way OCA2 and HERC2 operate in human eye color genetics. Variants in this gene and in the regulatory region surrounding it influence how much eumelanin gets deposited into the developing iris tissue during the weeks immediately after birth. Because here is something that most cat owners discover firsthand but rarely understand. Kittens are not born with their final eye color. Almost all domestics. Kittens are born with blue or blue-gray eyes regardless of what color their eyes will eventually become, because the melanocytes responsible for producing iris pigment have not yet fully activated at birth. The Final color does not stabilize until somewhere between 3 and 6 months of age, as the pigment-producing cells receive their developmental signals and begin depositing eumelanin at the rate their genome has. What happens during those 3 to 6 months of eye color development in a kitten is, at the cellular level, one of the more elegant biological processes in mammalian development. The cells responsible for producing eumelanin in the iris are called melanocytes, and they are not originally iris cells at all. They are migrants. During embryonic development, they travel to the iris from a structure called the neural crest, a temporary band of cells that forms along the developing spine of the embryo and then disperses outward to populate dozens of different tissues across the body. The same ancestral cell population that eventually becomes part of the nervous system, the facial bones, and the pigment cells of the skin and fur also sends a contingent to colonize the iris. When those melanocytes arrive and settle into the developing iris stroma, they carry with them the complete genetic instruction set that will determine how much pigment they eventually produce. But the instructions are not executed immediately. They wait for a developmental signal, a cascade of molecular triggers that begins shortly after birth and plays out over weeks, gradually activating the pigment production pathway to whatever level the genome has specified. In a cat whose genome carries the right combination of low expression variants across OLFML3 and the handful of modifier genes that fine-tune the final output, that activation plateau lands in a specific narrow range. Enough eumelanin to prevent the pure scattering effect that produces blue, not enough to overwhelm the scattering and produce gold or copper. The physics takes over from there and the eye reads as green. What makes this developmental window scientifically interesting beyond cat biology is that it serves as a remarkably clean natural experiment for understanding how pigmentation gene expression is regulated in real time. Researchers studying feline eye color development have used it to probe questions about melanocyte activation timing, regulatory gene networks, and the sensitivity of color outcomes to small quantitative changes in gene expression that would be far harder to study in other biological systems. A 2021 paper in PLOS Genetics used the variation in domestic cat eye color as a model to investigate how polygenic traits controlled by multiple genes rather than a single switch, produce continuous ranges of outcomes rather than discrete categories. This polygenic complexity is part of what makes green such an interesting color to land on. Among domestic cats, eye color exists on a spectrum running from pale blue through various shades of green, gold, hazel, orange, and copper, with each point on that spectrum corresponding to a different eumelanin concentration in the iris stroma. Green occupies a specific zone on that spectrum, narrow enough to be visually distinctive, wide enough that there is genuine variation within what people call green. The pale, almost blue-green of some Russian blue cats is genetically and optically quite different from the vivid, saturated emerald green that appears in certain lines of Norwegian forest cats or the warm, slightly golden green seen in many mixed-breed domestic cats. All of these fall within the category a person would describe as green if asked, but they represent meaningfully different points on the underlying pigmentation spectrum, produced by different combinations of alleles across the relevant gene network. Documented population histories that trace back through very specific geographic and demographic bottlenecks. The Russian blue, whose distinctive blue-gray coat and vivid green eyes are among the most 19th century. The founding population of the breed, as it exists today, was almost certainly small, and the intense selective breeding programs established in Britain and later across Europe following the Second World War, during which the breed's numbers had been severely reduced, further compressed the genetic diversity of the population. The vivid green eyes that now appear as a breed standard requirement in Russian blues are not an ancient stable trait stretching back thousands of years. They are, in significant part, the genetic signature of a founder effect, a trait that happened to be present in the small group of animals from which the modern breed descends, and that was then amplified and fixed by selective breeding pressure across roughly 20 generations of deliberate human management. The Norwegian forest cat tells a related but distinct story. Genetic studies, including work published by researchers at Uppsala University, have suggested that Norwegian forest cats represent one of the older, semi-distinct domestic cat populations in Northern Europe, with ancestry that diverges from the broader Western European domestic cat population at a point that may correspond to the Viking Age or earlier. The population likely developed in geographic semi-isolation in Scandinavia, with selection pressures that included survival in a genuinely cold and demanding environment rather than the artificial aesthetic preferences that drive modern show breeding. The green eyes that appear commonly in the breed today were almost certainly present in the ancestral Scandinavian population from which it descends, carried forward not because anyone selected for green eyes specifically, but because the allele frequencies in that isolated northern population happened to produce green as a common outcome when the developmental pigmentation process ran its course. This geography matters enormously when you start asking where the green eye alleles themselves originally came from. Because domestic cats spread out of the Near East in those 2 documented waves, the ancestral source of the pigmentation gene variants that produce green eyes in cats today almost certainly lies somewhere in that original Near Eastern founding population. Researchers working with the ancient cat DNA dataset from the 2017 Nature Ecology and Evolution study were not able to reconstruct eye color directly from the oldest specimens due to DNA degradation, but the population genetic modeling in that study supports the inference that pigmentation variation, including the variants associated with reduced eumelanin expression, was already present in the Near Eastern founding population before the first dispersal wave began. Green eyes There's a phenomenon in green-eyed cats that veterinary ophthalmologists encounter regularly, but that most cat owners never think to ask about, and it connects directly to one of the deeper questions in feline pigmentation genetics. Look carefully at the eye of a green-eyed cat in different lighting conditions, and you will notice something that parallels what happens in Hazel human eyes, though for entirely different structural reasons. In low light, as the pupil dilates and opens into that distinctive vertical ellipse that is one of the signature features of feline visual anatomy, the visible green of the iris appears to intensify, to become more saturated and vivid. In bright light, as the pupil contracts to a narrow slit, the green can shift toward a cooler, slightly more blue tone shade. This is not imagination, and it is not simply an artifact of the ambient light color in the room. It is a geometrical consequence of how much iris tissue is actually visible to an observer at any given moment, combined with how the angle of incoming light interacts with the layered structure of the stroma. When the pupil is wide, the observer sees more total iris surface area, and the scattering effect that produces the green color has more tissue to work across, producing a richer, more saturated apparent color. When the pupil is narrow, the geometry of the visible iris changes, and the angle at which light enters and exits the tissue shifts in ways that subtly alter the balance between scattering and absorption. These have been so disproportionately represented in mythology, folklore, and cultural imagery across so many different human societies. An eye that appears to change with the light, that seems to shift and intensify, depending on conditions, reads to human observers as alive in a way that a fixed, static color does not. The cultural fascination with green-eyed cats is not purely aesthetic. It is, at least partly, a response to genuine and observable optical behavior that the human visual system interprets as expressive and dynamic. Ancient Egypt is the most famous chapter of the human relationship with cats, and it is worth examining carefully because the popular version of that story contains a significant genetic misconception. The cats venerated in ancient Egypt and depicted in the extraordinary surviving artwork from the New Kingdom period onward were not the ancestors of today's green-eyed breeds in any direct or exclusive sense. The sacred cats of Egypt, associated with the goddess Bastet and kept in temple precincts from at least 2000 BCE onward, were descended from the African wildcat Felis libica, the same subspecies that gave rise to all domestic cats globally. But the eye colors depicted in Egyptian cat sculpture and painting are overwhelmingly gold and amber, not green. The green-eyed domestic cat, as a common and recognizable type, appears to have become significantly more prevalent after the second dispersal wave, the one that moved cats across the Mediterranean world via maritime trade routes, rather than being a defining feature of the Egyptian founding population. This is a meaningful distinction because it suggests that the allele combinations producing green eyes became more common in the global domestic cat population, not through the earliest phases of domestication, but through the specific population mixing events that occurred as cats moved out of their original Near Eastern range and encountered the founder effects of new geographic. The science of what happens to a small founding population's genetic variation when it moves into a new environment and expands is called founder effect genetics, and it has been one of the most powerful explanatory frameworks in evolutionary biology since the mid-20th century. When a small group of individuals breaks away from a larger ancestral population and establishes itself somewhere new, it carries only a random subset of the total genetic variation present in the source population. By chance, some rare variants that were uncommon in the original population may be overrepresented in the founders, and therefore become significantly more common in the descendant population. Other variants that were common in the source population may be absent from the founders entirely, and effectively disappear from the new lineage. Applied to feline eye color, this framework predicts exactly what the genomic evidence suggests. In certain geographic regions where domestic cat populations were established by small founding groups moving along, specific trade or migration routes, pigmentation allele frequencies would have drifted away from the Near Eastern source distribution, producing regional populations where green eyes were more or less common than in the ancestral population simply because of which cats happened to be on the boat or in the grain cart that founded the local lineage. Researchers at the University of Leuven published work in 2020 examining mitochondrial DNA variation in domestic cat populations across Europe and North Africa, finding clear population structure that reflects exactly these historical dispersal roots. Northern European cat populations show measurably different genetic signatures from Mediterranean populations, which in turn differ from Near Eastern populations, in patterns that closely track the known historical geography of human trade networks. The green eye allele frequencies in these regional populations are not randomly distributed across that structure. They cluster in ways that suggest green eyes became particularly common in certain northern and Western European lineages during the medieval period, possibly through a combination of founder effects during the expansion of cat populations northward with monasteries and trading posts, and potentially through some degree of deliberate or unconscious human preference for certain coat and eye color combinations in the cats that were kept close to human settlements versus those that lived more peripherally. That last point opens a genuinely unresolved question in the field. Was there any human selective pressure on cat eye color during the pre-modern period before the formal establishment of cat fancy organizations and breed standards in the 19th century? Later due with extraordinary deliberateness. But the absence of documentation is not the same as the absence of preference. Cats that were kept as household companions, that were valued for their temperament and appearance as well as their rodent hunting utility, may well have been selected informally for visual characteristics that appealed to their human owners, including eye color, without anyone writing down that selection criteria. The genomic signature of such informal selection, if it occurred, would be very difficult to distinguish from the signature of neutral genetic drift in small founder populations. There is one more layer to the green eye story in cats. Any functional advantage to the animals carrying them, or are they simply a beautiful accident? A byproduct of demographic history that persisted because nothing about them was harmful enough to be selected against. This question has generated genuine scientific disagreement, and the competing answers reveal something important about how evolution actually works in domesticated species living alongside human beings. The functional biology of the feline eye is, by any reasonable measure, extraordinary independent of color. Cats possess a structure behind the retina called the tapetum lucidum, a mirror-like reflective layer that bounces light back through the direct functional sense. So the question of whether green eyes specifically offer any visual advantage over gold, copper, or blue eyes in cats is a separate question from the general question of whether cat eyes are visually sophisticated, and the answer to this specific question is considerably less clear than the answer to the general one. Some researchers have proposed that moderate iris pigmentation levels, the same range that produces green in eyes, may offer a marginal advantage in certain lighting environments by reducing internal light scatter within the eye itself. A densely pigmented dark iris absorbs stray light effectively, but may slightly reduce overall light transmission in very dim conditions. A very lightly pigmented iris allows maximum light transmission, but may produce more internal scatter in bright conditions, slightly reducing contrast and acuity. A moderate pigmentation questioned and concluded that while the theoretical basis for a moderate pigmentation advantage is plausible, the empirical evidence in domestic cats specifically is insufficient to confirm it. The honest scientific position is that green eyes in cats are probably not strongly selected for on functional visual grounds and are better understood as the stable output of a specific pigmentation genotype that causes no disadvantage and therefore faces no elimination pressure. So what does appear to be real, and what several researchers have pointed to as a potentially underappreciated factor in the prevalence of green eyes in domestic cat populations, is the role of human aesthetic preference operating as an informal selective force over thousands of years of close cohabitation. Humans have strong and well-documented visual responses to certain eye characteristics across species. Studies in human psychology have consistently shown that lighter, more vividly colored eyes in companion animals attract higher ratings of attractiveness and perceived intelligence from human observers than darker eyes, though the effect sizes vary and the research has important limitations. If even a modest human preference for green or light-colored eyes in cats translated into slightly better care, slightly higher rates of indoor keeping, slightly better nutrition, or slightly higher likelihood of being allowed to reproduce rather than being driven away from a settlement, that preference would constitute a real selective pressure operating across hundreds of cat generations. Across 10,000 years of domestication, even a very small, consistent bias in human behavior toward green-eyed cats could have meaningfully shifted allele frequencies in populations living in close human proximity compared to feral populations living at greater distance from human settlements. This is speculative, and researchers working in this area are careful to say so. But it is not empty speculation. It is a hypothesis grounded in well-established principles of how human preferences have shaped the evolutionary trajectories of domesticated species across every domain from agriculture to companion animal breeding. The dog genome carries hundreds of signatures of human-driven selection for behavioral and physical traits. The domestic cat genome carries far fewer such signatures, consistent with the less intensive management history, but the signatures that are present cluster around traits that matter to human observers. Whether eye color is among them remains an open and genuinely interesting research question. Ancient DNA methodology has advanced rapidly enough in the last decade that researchers are now beginning to reconstruct pigmentation genotypes from archaeological cat remains with meaningful resolution in the ancient and medieval world. A collaborative project currently underway between researchers at the Center for Geogenetics in Copenhagen and several partner institutions is building a genomic dataset from archaeological cat specimens spanning the full geographic range of early domestic cat dispersal, with pigmentation loci among the specific targets for reconstruction. The preliminary results, presented at a 2023 conference on ancient DNA, but not yet fully published at the time of writing, suggest that the allele frequency shift toward lighter eye colors in European domestic cat populations occurred significantly later than the initial domestication event, consistent with the founder effect and post-dispersal drift hypothesis, rather than with selection operating in the original Near Eastern founding population. What all of this means, taken together, is that the green eyes looking back at you from your cat's face are carrying a history that spans continents and millennia. They are the visible output of a pigmentation gene, network that was already present in its essential form in the wild cats of the ancient Near East before the first grain stores were ever built. They were shaped by the demographic accidents of small founding populations, moving along trade routes across the ancient Mediterranean world. They were amplified in certain lineages by the deliberate and intensive selective breeding programs of the 19th and 20th centuries, and they have been maintained across all of those thousands of years, at least in part, by something as simple and as profound as the fact that human beings have always found them beautiful. That last point deserves a moment of genuine reflection, because it is not a trivial observation. The relationship between domestic cats and human beings is one of the longest and most consequential interspecies partnerships in the history of this planet, and it has been maintained not primarily through utility, the way the relationship with cattle or horses was maintained, but through something closer to mutual aesthetic appreciation. Cats chose proximity to humans because humans provided resources. Humans chose proximity to cats because cats provided pest control, yes, but also because humans found them visually and behaviorally compelling. The green eyes that emerged from that partnership through the specific genetic pathways this story has traced, from neural crest cell migration through founder effect dispersal to Victorian breed standardization, are in a meaningful sense a joint product of two species' evolutionary histories running alongside each other for long enough to become genuinely entangled. So the next time you look into a pair of green cat eyes, understand that you are not looking at a simple biological feature that can be fully explained by any single mechanism. You are looking at the output of a developmental process that begins before birth and completes itself over months. You are looking at a physical illusion generated fresh by the laws of optics every time light enters that iris. You are looking at the 10 millennia of human beings finding exactly that color in exactly that eye, impossible to look away from. That is not a small thing. That is the full, complicated, scientifically documented story of where those eyes really came from. And it begins and ends, as so many of the deepest biological stories do, not with a single dramatic moment of mutation or discovery, but with a long, patient accumulation of history doing what history has always done, moving populations across landscapes, bringing different lineages into contact, and leaving the evidence written in the tissues of every living thing that survived the journey. If this